which is another of these major research environments, also based in biology here in Lund, the Centre for Animal Movement Research. Now, important point about this meeting that it's actually being streamed directly to YouTube. So if any of you have problems with the authorities or feel that you don't want to be on film, now's your chance to leave. The whole point of this meeting, it started spontaneously when some of us were sitting, when one has a little bit more time in the summer, to sit over coffee and discuss with the few people that are still available in the department. And this meeting was actually born from a discussion around the coffee table. And it feels really nice to have so easily and without any fuss managed to end up in this situation where we have some of the really leading proponents, practitioners within citizen science here today. And the idea of this meeting is both to inspire and inform and lead to some hopefully good discussions. It's going to be rather strictly divided into two sections, and I want to try and keep these two sections separate because citizen science covers a mass of different things. So in the morning we're going to be focusing on inspiration, teaching, lifelong learning, species identification skills, and sharing information with other people. And then in the afternoon, we're going to change focus a bit and focus on data collection and the analysis of data collected by non-academic natural historians. So what goes in, what can we get out? Each of the sessions will be ended, if we keep to time, by a slightly longer discussion. And if you can be disciplined in the morning, ask questions that have to do with the topics in the morning session, and in the afternoon, focus on the afternoon's topics. So that the morning speakers, right at the end, they can come forwards and you can all ask questions. And then also the speakers have other possible topics for discussion if we run out of steam or if the topics haven't been taken up already. A quick information about the streaming. If there's time for questions after the individual talks, can you please make sure that you talk clearly and loudly? Because otherwise we'll have to fiddle around with the microphone for the questions. And it would be nice to avoid having to do that. Coffee and lunch, practical information. All the people who are registered here have got coffee and lunch arranged. Other people who are here without being registered, go to the back of the queue, and if there's something left over, you're welcome to take it. And there probably will be enough for everybody. So, without any ado, I'm going to introduce our first speaker, who many of you will have heard of, Jonathan Silvertown from the Open University in the UK. I'm going to...
Thank you very much, Anna. It's extremely brave of you, actually, to invite me here, because last time I was here was in 2010, and well, I gave a talk. The Earth moved, a large volcano in Iceland erupted, and all of European airspace was closed. So that's quite a risk you've taken in doing this. I'm going to be talking about... I'm going to be talking about amateur natural history, really, and in particular, the task of identification. Identification is absolutely key to everything we do as ecologists and evolutionary biologists or natural historians, however you want to describe yourself, because if you don't have the name, you cannot do all the other stuff. You can't Google something if you're a student, or discuss it, or put it in a project. I mean, it's kind of obvious. It's very fundamental, and certainly in Britain and many parts of the world, we don't actually teach our students how to identify stuff, and haven't done for over 50 years. So how do we actually give students and others, members of the public, those essential fundamental skills that they need in order to do everything else? So I'm rather pompously describing this as a new shape for natural history because there are new opportunities which I'm going to tell you about. Obviously, once you have got an identification of something, you can then, oops, interesting note, you can then create data. The process of identifying is actually a process of learning as well, and as a result of that, you can then identify other things that look the same or that you think look the same and essentially determine the relationship among related species. More data can also be used to help with identification, and I'll show you how we do that in a project called iSpot. And that's essentially the structure of this talk, and I'm going to go through those different areas and show you how they're all linked up. So iSpot is a website. You'll find it at ispotnature.org, which is essentially designed to crowdsource identification for anybody. Anybody can use it, anybody can suggest a name, and it's designed to do that three-stage process that I just showed you, to help people get identifications, to learn how to do it, and then to generate data. And we do it by working with a lot of organizations. This is just about 90 of the 150 organizations that we work with, mostly in the UK and in South Africa, where we have another website doing essentially the same thing. These are very specialized. Here's the Hoverfly recording scheme. Somewhere here, Helen is the Ladybird recording scheme, and so on. So over the last 5 years that we've been working, we've built up this network of people who are interested in particular groups of animals and plants, or work in particular areas, to help us run this crowdsourcing website. So this is what it looks like if you visit it. You can also find it on Android phones as an app. And we'll shortly be producing a responsive design that will enable you to use it on all mobile devices. So these are observations that people have submitted. They're coming in all the time. And here's basically just a selection of things that I took from iSpot a while back, and these are our current statistics. So we have 330,000 observations, over 500,000 images, nearly 40,000 registered. In fact, it has now reached 40,000 registered users. And we've identified 30,000, or say we, the social network, has identified 30,000 species by this method. And I'll show you how it works. So here's a map. Everything is geo-referenced. This is the distribution of observations a few days ago around here. And iSpot deals with all species. That's really important, that all taxa are included. It's a one-stop shop. And you'll see here that birds, which I know a lot of people in the room are especially interested in, comprise 17% of observations. I think in Arc Patalan it's 75%, somebody told me yesterday. So we very much focus on all taxa. There are even a few bacteria in iSpot, although identifying them isn't that easy. And that was just the observations that were received in that period there at the beginning of last year. We provide data downloads to the schemes and societies that I mentioned earlier. So although iSpot is designed to help people put names to things, of course in the process they generate data which are then available for scientific and conservation purposes. Here's an example of what you can see in the data if you look with the eye of knowledge. So this is a data set, one of our early data sets we gave to an expert in shield bugs. That's the distribution of 300 observations. It's kind of slightly cut off by the bottom of the screen. But anyway, that we downloaded, or we gave. And these are the notes that we were sent back by the expert. He said this was the first record of this species on the NBN Gateway, which is the national repository for biodiversity data in Britain. an intriguing record at the Firebug in London. I guess intriguing means you haven't seen it often before in London. Multiple records of this thing, which is new. I think it lives on conifers. And these species were known to be moving northwards in response to climate change. And you can see those patterns within the data in iSpot, which of course is just produced by people using iSpot. We don't actually direct them to go out and look for things. This isn't a survey operation. It's very much casual records, I suppose you could call them. And this is how the identification process works. So you upload a photograph. Here's a moth that somebody's put up. And that's the location. If you were to click on that link, you'd see a Google map which would show exactly where it is. Unless it's rare, in which case you can obfuscate obscure the exact location, which is very important, particularly in South Africa, where you have things like white rhino, which you really mustn't locate precisely, or for obvious reasons. And so the guy who put this on reckoned it was this species of red carpet. You see here the name of the person who suggested this name. Might not actually be the person who put the observation into iSpot. It could be somebody else. In this case, it is the same person. And that indicates their reputation, their earned reputation, for invertebrate identification. Nonetheless, despite that's actually quite a high reputation, despite having a high reputation, somebody else has suggested a different name. And it turns out that quite a few people agree with them. And here you can see the usernames of the people who agree, their earned reputations for the relevant taxon, their affiliations to the various organizations that I showed you in the second slide. And what iSpot does is it calculates the reputational weight between that name and this one, and then says, okay, this is the likely ID, because many more people with greater reputation think it's that as opposed to that. And when the person who suggested this name actually has agreement from others, his or her reputation is incremented. So it bootstraps. And over time, as you suggest names that are correct, according to others who know, then your reputation rises, and the reputation you can give others when you agree with them also rises. You do not get reputation for agreeing. So you can't enter into a sort of coalition with other members and say, I'll agree with you if you agree with me, and your reputation goes up. That wouldn't work. So we've carefully designed this so it cannot be gamed. And as far as I know, nobody's managed to crack it yet, which is good. Here is an example of a mystery user, me, and the groups and what somebody's reputation looks like. So that's everything in iSpot is public. You can see that if you're logged in. And in fact, you can see all content in iSpot apart from this without being logged in. So this is how it works. What I've done here is I've taken 100,000 observations, which were submitted to iSpot with no name, no species name. And this is how long it takes for them for those observations to acquire a name. This is time along here, first 10 minutes, first half an hour, the first hour, and the height of the bar is essentially the proportion of observations cumulatively identified within this period. So 56% of this sample of 100,000 observations were identified within 1 hour. Over half of the observations are identified within an hour, and it can happen, well, here's a quarter of them are actually identified within 10 minutes. It can happen within seconds. It's extraordinary. There are people out there who are just waiting to see interesting stuff so they can provide names. People love doing this kind of thing, and out of that we generate an extremely useful service that helps people learn. When we started iSpot, and I think we were among the first to do this anywhere, it really wasn't clear it would work. Fortunately, that didn't occur to the people who funded us, and fortunately for both of us, it actually works extremely well. One thing we didn't know was whether it would scale. So we started off by getting lots of experts involved. So iSpot's a network of experts, beginners, and everybody in between, and people of all ages, I should say. So school kids use this, university students use it. It was quite possible that as iSpot grew, we would exhaust the capacity of the experts to provide answers. This graph shows that that hasn't happened, and that, if anything, iSpot has got better at doing its core job of putting names to things. So the blue bars are the number of observations submitted per month from roughly when we started. We started a bit earlier than this, but in 2010, and this is the peak of last summer. In South Africa, you'd see a mirror image of this because, of course, it's in the southern hemisphere. This is just the UK site. Sorry, I say mirror image, I mean these, yeah, just in terms of the time. And the red line shows you the proportion of observations that receive a likely ID. And you'll see it almost immediately went above 90%. And if anything, it's been climbing as the number of observations submitted per month has climbed as well. So iSpot is scalable. It appears it can work at any scale. It doesn't matter how many observations we get in. And also it works in areas where there's mega diversity. It works in South Africa where diversity is very high. I'll come back to that in a minute. So this is what actually is going on. This is a rather faded picture of the British Isles with a visualized social network. This is the social network around one observer who has submitted in this picture a sample of 506. This is a sample of 506 of his observations. And the blue dot shows where he made the observation, and the red star shows the location of the person who identified that observation for him. And the yellow line, of course, joins the two. So this visualizes the social network around one user, who's quite a busy user. I mean, this is the top end of people using iSpot. There are people with thousands of observations, but 500 is pretty good. And okay, this is one user, and we've got 40,000 of them. Right? So that gives you some idea about what's going on within iSpot. Of course, there aren't 40,000 people as busy as this, but even imagine 20,000 of those laid on top of each other. So it's quite a remarkable network. And as a result, we've discovered species new to Britain. Here's a moth, which a little girl of 6 discovered and was identified on iSpot within 24 hours. It now sits in the Natural History Museum, and it's the first specimen of that species to be seen in Europe. Interesting because actually elsewhere in the world it's invasive. Here's a new bee fly species new to Britain. And we pretty much daily find new records of rare things in South Africa where there's very high rates of endemism, and they're always finding new populations of red data book species because people are just out there and submitting stuff to iSpot. So this is the global distribution of iSpot observations as of about a month ago. We actually have the only 3 angiosperm species in Antarctica included. We didn't set out to do that, by the way. It just happens. And obviously we're particularly busy in this part of Europe, and particularly in the UK. There's even more observations in South Africa than that shows. It really should just cover that whole part of Southern Africa. We're just about to launch in Hong Kong over here. We've opened up in Chile recently. And the way we work is we like to have local communities, because that kind of set of organisations I showed you earlier is essential to getting things going. Once it gets going, it keeps going. So, but we don't want people being disappointed by putting things in and then not getting an answer. Getting an answer depends upon having local expertise. So that's how we work. Here's just a little snapshot of the South African data. Because another thing we didn't know is, okay, it works in Britain where we have a limited number of species, but would it work in a megadiverse country like South Africa with, in fact, they have 20,000 angiosperms. These are just the numbers that actually are on iSpot. So they've actually now, I think they've got about 18,000 species in iSpot that they've identified. Hundreds of mammals, hundreds of herps, and everything. They're now using it in the marine environment as well, so the numbers are going to rocket. And that's the equivalent, the South African equivalent of the picture I showed you in the UK, all the organisations they're working with. So this is a real network, and as well as an online network. These represent lots and lots of different people who go out and record stuff. And here's an interesting example of what can happen when you join people together like this. So this image came in from a clinic in Zululand, and it was posted saying some children in Zululand have eaten these seeds and are very ill. We need an ID if at all possible. And that was what was posted, just that with a 5 rand piece. 5 rand? 5 cent piece. 35 seconds later, somebody said, suggested, this is the physic nut tree. 2 people agreed. It became a likely ID. The identification was sent to the doctor who'd inquired, and the following day we got the message back saying, I'm happy to report that although these 5 kids were very ill, they've been discharged and they're fine now, and it's good that we now know what these seeds are, because this isn't the first time this has happened, and it's probably going to happen again, so it's good to know what we're up against. So, you know, the problem was in one part of South Africa, the people with the knowledge were in another, and the whole thing happened through iSpot, which is hosted in Europe. So that just kind of, you know, reinforces the amazing power of social networking. I referred to this earlier. Here's an example of what you do when you have a species that people love to see, but of course, you know, rhino is now being poached with machine guns, and people get killed in the process of defending or trying to poach these animals for the horn. So you really don't want to tell people where they are, and so what we do is we put we put the observation somewhere inside a box. So if you were to do that, it does not tell you where the rhino was. All you can say is it's somewhere in the Kruger Park, and people know that. That's not a big secret. So we automatically hide red data list species that are submitted and identified on iSpot as a way of protecting that. And recently we started stripping out the EXIF data so people can't download the photograph and then discover where it was taken in a different way. We also use iSpot for invasive species surveillance. Here's an example. The Western Cape of South Africa is a major centre for, in fact, the centre for evolution of the genus Erica. There are over 800 species there, many of them in Table Mountain National Park. And somebody submitted this and said, it's very similar to an Erica I photographed in Wales. I've seen it on this path, can you tell me what it is? And they put down Erica, and very soon afterwards, I think, what was it? Okay, that afternoon, somebody said, actually it's Calluna vulgaris, which is a serious invasive in the southern hemisphere. It's a very serious problem in New Zealand, and finding Calluna in the centre of diversity for Erica is potentially a big problem. So that's where it was, and underneath each observation there's a kind of comments stream, and so they sent out a team to try and find it and eradicate it. Here is our latest branch of iSpot, so to speak. It's all on the same website. This is in Chile. The Spanish translation actually isn't complete yet, so you won't see that in Spanish at the moment, but probably by the end of May it will be there in Spanish. Here's something, I went to Chile to start this off, and somebody discovered a chiton that hadn't been seen for 70 years. He was an expert in chitons, so it wasn't a chance thing, but and that appeared on iSpot, which is really nice. And then you can share it with your students in this case. So it's being used a lot by university students in Chile at the moment. That's the chiton. Another thing we do in iSpot is we look at ecological interaction. So I'm an ecologist, and from the very beginning, I wanted to show people what the relationships were or are between species. And so what we've done is, courtesy of the Biological Records Centre in England, we have a big table which tells us which plants are eaten by which insects and which insects plants eat. And so when in iSpot you identify one of these insects, for example, or any herbivorous insect actually, we can show at the bottom of the page a little list of, not a list, a carousel of pictures of the plants that it eats, and vice versa. So when you see the plants, we'll show you all the insects that it eats, illustrated by pictures submitted by other users. So iSpot contains only, in the jargon, user-generated content. We haven't actually added photographs of anything, sort of library pictures. So you can then actually drill down, you can click on a picture and find out where it was seen and what people said about it and all this sort of thing. So this is sort of first-hand information, but then gathered together to produce ecological relationships like this. And we also now record new ecological relationships that people have seen. So if you see an insect visiting a flower, you can record the insect, the flower, and the relationship on iSpot, and it then at some point when we will be feeding the data back to the Biological Records Centre. In South Africa, we're essentially doing the same thing. They don't have this, we haven't digitised the information that exists about insect-plant relationships, but we can and will do that at some point. So now we're beginning to expand beyond just identifying things to help people understand the relationships, the ecological relationships that of course, are everywhere. That's just an example. This is black poplar, and these are the species illustrated with iSpot material, iSpot photographs that are known to feed on black poplar. So I've said all along that iSpot's about learning, and so we actually, how do we know that people are learning? This is really important, that we know that people learn. I've shown you the remarkable evidence of that iSpot works in terms of putting names to things. Over 90% of observations get a name, and they get names very quickly. But are people learning in the process? And that turns out to be actually quite a hard thing to demonstrate. So this is our first stab at doing it, and I'll show you another way in a minute. So what we did was we took 400 observers roughly, who'd made at least 50 observations each, and we then from that sample we said what portion of the observations were identified by the person submitting them, if it was their first, their second, their 10th, or their 50th observation. The idea being that the longer you've been observing, the more if you're learning, you should be more likely to put a name to it yourself rather than wait for somebody else to do it. And actually, this is what it shows. So basically, about 37% of observations, when first put on iSpot, the very first one that somebody puts on iSpot, are given a name by the person submitting them, and by your 50th observation, you're likely to be up in around somewhere around 60%. So you've nearly doubled the likelihood that you will at least have an attempt at naming it if you've been using iSpot for quite a while. This doesn't prove it's learning, but it's quite suggestive. And so actually what we've done in order to, to again use our data more effectively and to find out if people are learning, is we now have a quiz. So this is generated again from data in iSpot, and you can choose which taxon you want to be quizzed on, what level you can personalise it so that the quiz material focuses on the things that you've yourself put into iSpot or you've interacted with in some way. We have a record of everybody's activity, and we can use that to personalise a quiz. And here's an example of one kind of question. So select the image that contains the common read. And the nice thing about this is we record everything that goes on in this quiz, including wrong answers and how long it takes somebody to make a response and all that kind of thing. Because the wrong answers are actually going to be really useful to us in creating a new tool to help people identify things. I mean, this is the common read. Now, if people regularly misidentify that as the common read, what we can do is we can say, we will know statistically how often that happens, and then when people identify the common read in iSpot generally, we can say, okay, well 10% of the time, when people say they've got the common read, they've actually got this. So here's a picture of both of them to help you decide which you've got. So we can generate some extremely valuable data that can be used in creating a new ID tool just from the wrong answers in a quiz. We can also do that using the alternative identifications, like the one I showed you for the moth. So we can say, okay, well, people confuse the red carpet with the broad bar, whatever it is, and here's a picture of both of them. So we haven't done that yet, we haven't used that yet, but as the data increase in iSpot, that's the kind of thing that we can do. We also have Bayesian keys in iSpot. These are what you might call multi-access keys, I suppose, in the days of paper. The way they work are that we have them for a whole range of different taxa, and if you're interested in creating one of these, talk to me afterwards. We can do it very easily, put it online for you. So you have a set of characters. They're presented in the order with which, in the order that is most discriminating. So in this particular case, this key to woodlouse, the colour of the woodlouse is the most discriminating thing among the species in the key. You then select the best description of your specimen, and what that does is it reorders this list with the most likely thing at the top, but all the other things are still shown, and then it illustrates that. And this illustration comes from iSpot material. So if you've ever tried to produce a key and illustrate it, you will know how difficult it is to find good illustrations for the rarer species, for example. In iSpot, when people sign up, they give permission for their photographs and so on to be used in iSpot. So because all this is in iSpot, you can basically use any iSpot content in your key, which turns out to be very valuable. And then you can click on the name. They're all hyperlinked to get a description. Down here are the characters used so far. You can change your mind and so on. And in one or two cases, our keys have been tested against the dichotomous equivalent, and this key is a lot quicker. A few clicks will get you to the right answer, and with less prior knowledge as well. So this is certainly the way to go. And the software is generic, so we can generate a key like that from a basic matrix of species type characters. Another thing we do to help beginners is, again with user-generated content, is we just give people a gallery of the species that are most often seen in iSpot within different taxa. So here are examples of white waterfowl, and the top left, which is the mallard duck, is the thing that's most often seen in this group. And then you can sort of explore the gallery. Well, there it is. And you can explore the gallery. Here are the most commonly seen passerines. As a beginner, this is actually incredibly useful. Most books will organise things in terms of taxon, and you can flip through the book, but it's actually not terribly helpful to see the rare and the common things all together. This is basically ordered in terms of what's most commonly seen on iSpot in your area. So hopefully it should be a lot more useful. And then thrushes and so I'm now getting towards the end, you'll be glad to hear, Honor. We're continually developing iSpot. This is something we haven't released yet, but I thought I'd just show you what's coming. This is actually inside a new design for iSpot. You'll see this looks somewhat different to the slides I've shown you already. And this is projects, and what projects allows you to do is draw a polygon on a map and say, I'm going to look for all the birds in this wood, or all the fungi, or everything in this wood. And that polygon could be as big as you like, or as small as you like, and then you share it with people. And iSpot automatically retrieves all the relevant observations in that polygon. So there are a whole set of other filters you can apply as well, right down to a species name. So you can say, I'm only interested in the Amanita muscaria in Highgate Wood, and iSpot will automatically populate this gallery with observations of that species in that place, and then you can say down here why you're interested, and other people can comment, and any observations that come in that meet those criteria will automatically be sent to this page. This is just the beginning, and we will develop this. But it links to software for inquiry learning at the Open University, so it's expressly designed for doing projects with students, but it can be done. It can be used in all sorts of other ways, and you can see the data on a map or a list, and so on. So that will be coming out again in about a month, I think. Let's revisit my three lozenge plan for the new shape of natural history. Data. Within iSpot we now have distributions, we have images, and we have interactions. And relating to ID, we've got keys, the species surfer for exploring what's in iSpot in a sort of combination of what's common and what within different taxa. There's a social network, and we're looking at ecological interactions. And so in the ways I've described, we're using distribution to help people with the keys. The interactions are being fed back from the data back to the ID pages. The images, of course, in species surfer help people identify stuff. The quiz I showed you helps test whether people are learning. And we will be using the data in the way I described to help with identification later on. And so on. So at this point, everything's beginning to join up in iSpot. It's only been possible because of the thousands of people who use iSpot, the knowledge that they have, and the large number of observations that we now have. Much of this we simply couldn't have done when we started because we didn't have the data. But now we have the data and all the relationships that lie behind those data. This is what's possible. So that's my suggestion as to how actually within iSpot, but also potentially out there on the web, we're beginning to see a new shape of natural history. Thank you very much.
Well, thank you very much, Jonathan, and impeccable timing, of course. I want to start by asking a really crass question. Who pays for all this?
That's not a crass question. That's a question that almost obsesses me. Well, actually, the answer is here. So we started off with a big grant from the Big Lottery Fund for England, which funded us for 5 years and was absolutely key to our foundation and everything I've shown you. Since then, we've had money from foundations. And from the British Ecological Society, for example, who funded the ecological interactions that I showed you. And at the moment, the Open University itself is underwriting iSpot. But this is a continual process of fundraising to keep it going. The longer-term plan is that that the organisations around the world who use the iSpot service will pay a subscription for it. So South African National Biodiversity Institute pay us an annual fee. They get a real bargain, of course. as you've seen, that huge numbers of users and observations come in as a result of using iSpot, and the same will be happening in other parts of the world. So that's actually what our plan, but we are far from economic sustainability at the moment, and so we depend greatly on the OU.
Kristen.
Follow-up question there. So what is your budget?
What is our budget? We have to cut our cloth according to our coat according to our cloth. So we were spending hundreds of thousands a year. We're now spending, a couple of £100,000 a year.
100,000.
£100,000, yes, yeah. If you take everything into account.
You work with open source software. Have you thought of making modules that would be possible to integrate in other, for example, Drupal environments, so you could interact with iSpot from other sites? And,
yes, we have an API. So we're moving towards a point where everything will be driven by APIs. So the APIs are used to drive the mobile app, but we will be probably actually driving the website itself in that way. So that's one way we can work with other sites. We have a widget which we haven't deployed yet, but which will enable people on other sites to see filtered iSpot content. So we're working on that kind of thing.
There's a lady there.
Yeah. Has there been any publications coming out of this?
Not yet. We're working on a paper which will describe a lot of the things I just told you about as an example of development. As far as I'm aware, the data we collect, it sounds like a lot, and I think it is a lot given how it was collected, but the National Biodiversity Network and the Biological Records Centre have hugely greater quantities of data. So when people actually use data for scientific purposes, they tend to download it in much greater quantities from GBIF, NBN, places like that. So to date, it hasn't been used as a main source, but one day it will be, I think.
Sven.
What proportion of species can identify to species level from pictures alone in different organism groups?
I can't answer that directly. We will be able to answer that. I take the point of the question, absolutely. The shield bugs that I showed you, apparently they can all be all the British ones can be identified from photographs, which is one reason why we chose that particular group as a demonstration. There are many other groups, as you imply in your question, for which that is not true, and you can only go to, bumblebees, for example, you can only go with photographs to to a selection of 2 or 3 species in some cases. Partly it depends on what people have photographed, so it's a complicated question to answer, but yeah, I take the point.
We've got time for one last question.
So you said in the end that distributional data was one of the things this collected as well, but you didn't touch too much on it in the talk. Have you been able to look at some of those patterns of where species are actually found and identified? If we just look at the UK, if there's holes in remote areas.
We can do that. One reason I haven't mentioned it is because there are much better. We have a relatively small sample size because we're basically about helping people put names to things. We haven't, people upload an observation at a time, but if they're doing biological recording per se, then they'll upload hundreds of observations at a time, and that gives you much better geographic spread. So, it's possible that a later speaker, Helen Roy, will talk about this in relation to ladybirds. That's why I haven't focused on it. One more question?
No.
All right.
We're heading for coffee.
Coffee.
So
Ask me at coffee.
There's a coffee break, and then then we're back here again promptly at 5 past. So, thank you very much.